From the Oregon State University Extension Service, this is Pollination, a podcast that tells the stories of researchers, land managers, and concerned citizens making bold strides to improve the health of pollinators. I'm your host, Dr. Adoni Melathopoulos, Assistant Professor in Pollinator Health in the Department of Horticulture. I had a lot of feedback from a show I did last month with John Ascher, who's an assistant professor in the Department of Biological Science at the National University in Singapore. In that show, Dr. Ascher made the case that in some really meaningful respects, we're on the cusp of making a transition from where birding was before the 1970s to what it looks like today in the world of bee identification. Well, we are real fortunate we're able to coax Dr. Ascher to come back on the show to talk a little bit more about how this works. And today he's going to do something really interesting. We're going to talk just briefly about some of these different digital platforms for regular people to get involved with bee identification and collecting data, but then we're going to focus in on records collected through iNaturalist here in Oregon, and we're going to go through the top 10 most popular bees that people have recorded here in the state. And there's going to be a link in the show notes, and you can follow along. This is really wonderful. We're going to have a world expert walk us through how to identify some of the most common bees in Oregon, but also some good tips on how to record that data if you're out with your phone. For example, in a national park where collecting is very difficult to do, you can take a picture and how that information will go forward to contribute to science and some of the best practices for collecting data. Very excited. Here we go. Dr. John Ascher today on pollination. Welcome back, Dr. Ascher. We're so excited that you agreed to come back on the show. There was a lot of interest in the last episode you did, and I think people wanted to know more about these online guides. So welcome back.
Okay, thanks for inviting me back. It was a great opportunity the first time, and I'm looking forward to getting into some more details about bees of the Pacific Northwest and Oregon and how we identify them and how we can use some of these online tools. So we'll be building on the end of our past discussion. And as we talked about before, there are now multiple citizen science portals that give us the opportunity to share our bee images, get them identified, and get them organized into projects and used by the public as a reference guide and many other things. So one of them is Bug Guide, and Bug Guide is an excellent place for sort of high quality cropped images that you want to share and get organized onto what is essentially a ever-growing online field guide. And for bees, this has been very successful, and many of the species, if not most, now have well fleshed out species pages with quite a bit of content, including links to distributional maps and so on and so forth. So this is a wonderful reference guide, and it's sort of a de facto field guide that you can refer to because it has a high quality of curation. And the tools are quite excellent to ensure high quality of image presentation, but also the curators and those people who are identifying the bees have a lot of direct control over how they appear on the pages. And so that leads to really good data quality.
Okay.
So I recommend that site. And then, of course, iNaturalist has been just growing exponentially and is probably the best place for a very high volume, a very high quantity of excellent images for all animals and plants, but certainly including bees. There are now more than 500,000 bee observations there, and these have been sent in by more than 100,000 observers, and there are
my goodness,
and there are almost getting close to 15,000 identifiers. So the amount of buy-in by the public and the amount of effort and work that goes into this is really stupendous.
Well, one of the things that prompted this episode is that you pulled down. You have a lot of these. There's a lot of ways to filter the data in iNaturalist, and you created a list of the bees of Oregon that that have been identified. And I was really shocked. There's 6,300 observations that people have collected and 116 species that are identified there. That is 116 species is really remarkable.
Yeah, I think maybe the most remarkable thing is, if you remember, when the last time we discussed, we briefly chatted about this, and the numbers have gone up very substantially just since a few months ago. So the most impressive thing about iNaturalist as far as bees are concerned is to me, is the rate of increase, where I used to be able to sort of keep up with the observations pretty well, but now that so many are coming in, there's been such a tremendous enthusiasm, not just in Oregon, but globally, that these numbers just keep jumping up. So globally, there are now more than 2,000, actually 2,370 identified species. So more than 10% of the world's bees are of known bee species are represented already in this resource globally, which is quite a lot, because the status quo, or the received wisdom, was that bees are one of these groups that are too difficult to identify from images, and that it's strictly necessary to have rigorous specimen collection and identification at all times. But what I think is happening over the world is that things are entering a new era where it's more like birding. And just as in birding, they went from a case where nobody could identify, say fall warblers, and then they figured that out, and then they moved on to the gulls, and then they moved on to the immature gulls, and now they're working on the hybrids and really the most difficult cases. And I think in the case of bees, we've seen a situation where the really obvious bumblebees that are the usual color form were maybe sorted out years ago. But just now, I think we're grappling with a lot of these really difficult identification problems, regional variation, photo artifacts. All of these phenomena are getting understood much better, and also the quality of the photos gets better and better. And the quality of the initial identifications gets better as the user community builds their own expertise. So things are accelerating, and we're able to assemble a range of species and a quantity of observations and a percentage of coverage of how many things can be identified that I think are unprecedented and that wouldn't have been anticipated. I think people in general have been too skeptical, in the sense that I think that looking back at some statements people made, they would say, well, bees, sometimes you can identify them to genus, but usually that's even difficult, and getting to species is usually impossible. But if you look at places like Germany or England, or areas where they really know the bee fauna, they can identify a large proportion of their species, the top experts. And I think things are moving that way in the United States as well.
Well, I was going to say that you're one of a few taxonomists have really jumped, at least with the North American bees, and likely many other areas of identifying on iNaturalist. So I know I remember I was in Mexico City a couple years ago, and I saw Trigona, and I thought, oh, I've never seen one of these again. I put it on iNaturalist, and the next day I saw it was you had taken a look at it and verified it. So sounds like you're really invested. Like you've seen the potential in this technology. Can you tell us how taxonomists interact with things like iNaturalist and these other portals? Because it must be it's a little bit more fuzzier than having a specimen in front of you, but you probably also know a lot of these common things really well.
Yeah, so it's a bit complicated, as you can imagine. I mean, with this number of this many records and this many, I would say that I'm sort of looking for a couple of things. One is the low-hanging fruit. So how can I help the community and also help myself by just getting the obvious things out of the way, the honeybees cleared out. So I'm trying to get more and more efficient methods of using the various filters, like they have the identify tool, filtering that efficiently, moving forward very efficiently, and getting the obvious things out of the way. And then for the very difficult ones, I'm also trying to get an efficient methodology. So for example, I have an array of European guides covering all the European species. So in that fauna that I don't know as well personally as the North American fauna, I will keep by my computer an array of sort of a stack by this point of many different guides, for Germany and Czech and UK and France and all these different countries in Spain. And I have them ready as needed to consult. So
how many everybody wants to know, like, roughly how many digital vouchers have you determined? It's got to be
for all creatures across all the sites. I would say more than 1 million.
Man!
But I mean, that would also include specimen records. But anyway, for iNaturalist alone, it's about 285,000 bee observations.
Wow. Okay.
But I've also done quite a bit more at Bug Guide because I've been working at that site longer than iNaturalist. So one thing I want to get at is that these sites are actually quite well linked to each other, both conceptually in terms of overlapping communities.
Yeah.
So the idea that they're fully separate is maybe a bit false. I think they all have their strengths, and they're communicating with each other in the sense that Bug Guide is functioning as a very effective reference guide. And iNaturalist is an extremely good way to accumulate the vast number of records very quickly and reliably, and in a user-friendly way that really prioritizes the user experience or the contributor's experience. And then in other sites like Discover Life, things are sort of synthesized in a different way because it's more of a aggregating of the data and integrator of data. So, for example, the Bug Guide, not the Bug Guide, but the citizen science records can be contributed to these global biodiversity initiatives, and then they can be integrated with the specimen records and put up on maps. So we can, so the maps on the site of Discover Life would then include, in addition to specimen records, also the public image records from these other sites. So they're not entirely separate from each other.
All right.
And also the site, the records, the new state records and new country records and so on that I'm obtaining from a site like iNaturalist then go to update my files for state and country and provincial records, which then I share with Discover Life for their bee species guide and world checklist.
Fantastic. Well, let's move on a little bit to this question. What is best practices? So we have lots of listeners who are contributing. We were looking at some of the contributors here in Oregon. People, we all know. What are some of the best practices for contributing bee data on iNaturalist? What should people, what things should people be making sure to do, both in terms of pictures, but also in terms of it gives you that option of identifying the bee and, how far down that rabbit hole do you want to go?
Yeah, of course, there's no one size fits all answer to this, but I would say, first of all, take the best photo that you can. And people often ask, what is the best angle? And usually the answer is multiple angles, because very often, even if the photo is a bit blurry or low quality or uncropped, if you have multiple angles, often it's easier to identify the bee. So I would say don't take one photo of the bee, especially if it's a resting bee, like a sleeping bee. Try to get different viewpoints, including the tail, not just the head. Maybe an oblique view from the bottom in addition to the top, and things like this. So
yeah, because iNaturalist will allow you to add to that record. You can add as many pictures as you want.
Right, but that said, I would say be really careful that you actually have the same individual. If in doubt, it's better to submit them as separate observations. So one pitfall is a lot of people will sit and watch a bush and take photos of bumblebees as they arrive at the blueberry bush or whatever, and they'll end up taking 3 or 4 species of bees and then reporting them as a single observation.
Oh, right, okay.
If you're 100% sure it's the same bee, take multiple photos, link them, and submit them together, but try not to mix and create a virtual humbug, because that's not very helpful.
Okay. All right, so try to get multiple pictures of the same individual and from different angles. That sounds like a great tip. What else?
I would say you can make use of the iNaturalist artificial intelligence, but you need to be very cautious, because you need to realize there's a big geographic bias. So if you're working in an area where they have a huge number of observations, like maybe the Eastern United States or United Kingdom or somewhere like that, it might work well. But if you're in somewhere with like maybe Eastern Oregon, which has a smaller number of people, a lot of the bees there are not going to be within the system, and it's not going to work as effectively. So often,
right. So
automated.
Right. So when I'm in iNaturalist, I go, what did you see? If you press that, it starts to guess. It goes through and it says something. And you're saying that some of those may not even exist in your region.
Well, it's going to try to find a match. And so if a lot of the input data, if a lot of the data it's using to train the artificial intelligence is coming from the Eastern United States or from Europe or somewhere like that, then it might direct you towards a species which is not relevant. So the number one thing I would suggest to people is to try to verify that if you do identify to a low level like species or a precise level, try to verify that it's within the known or plausible range of the species.
Okay.
So the way you can do that is to go to a website like Discover Life or even iNaturalist itself and realize that there are probably going to be outliers and misidentified specimens that other people have submitted. But if you look at the general distributional pattern, if you're an outlier, or certainly if you're an extreme outlier, like if you're reporting a bee from the west where almost all the other records come from the east, then you might want to rethink that.
Well, and as you point out, like the very first thing, I just took a picture of a bumblebee on a screen, and the first thing it said is, we're pretty sure it's in this group. And so it said, I could just leave it at Bombus. And then, but then it has.
I would suggest leaving it at a general level if in doubt, because if you have it at Bombus and it turns out it's another genus, that doesn't really cause much trouble. The issue comes in if you try to identify a species, and then that can actually show up on species level maps and cause problems and also mislead other observers. So a lot of people will report Apis cerana, the Asian honeybee from North America, so it's constantly showing up on the maps.
Well, yeah. I suppose the other thing for you is if you go to a general identification, then it probably helps you in your filtering. You're like, I'm going to pull out the bumblebees, and then you're like, okay, and then you can go through and do your more finer work at that point.
Yeah, so it's a bit of a trade-off, because if somebody guesses and they're right, it actually expedites the process that they did that. Because if you guess at a certain level and it turns out that I when I'm checking the identifications, I can see what they guess, and it can actually expedite things. But if you make a uneducated guess, then it actually slows things down and complicates things and creates confusion along the way. So I would say, I would encourage people to make educated guesses that account for the distribution of the species, but don't make an uneducated guess, and don't accept the AI level at the species level necessarily. So the AI can point you in the right direction, but I think you as a human still need to make some judgments.
Okay, so that gets your records. We've got get the multiple images, different angles if possible, and really kind of go as general. Unless you're really sure, don't go down to the species as sort of best practices using iNaturalist.
Well, I think also like guessing at above the species level, like guessing at a genus or even a subgenus, doesn't really do too much harm. But when you guess at the species level, that can create problems, because then it shows up on those species maps, and that in turn can mislead other users. So I would be I would say stay away from making species identifications unless you have some solid basis to do it.
Well, that's great, and let's take a quick break, because that leads into our next section. We're going to look at the list of bees that are common here in Oregon on iNaturalist, and you'll kind of give us you'll help train our eyes a little bit towards what those different species are.
Okay, great.
Okay, we are back. So, Dr. Ascher, you've just shared a link with me, and this is really great. You can do the filtering in iNaturalist, and then you can create a report. And what you have for me are all the bee observations in Oregon ranked in terms of their observations. And I was really surprised with this. I would have thought honeybees would have been number one, but actually they're number two. Tell us a little bit, is it hard to tell honeybees apart from the other bees?
Well, it really can be remarkably difficult, especially for somebody who's not an expert. And probably the main reason for that is that honeybees are subject to a lot of color variability. So you get your classic honeybee, which is orange with black and yellowish stripes, but you can also get honeybees that essentially appear all black. And so those are often misidentified, or sometimes they're attributed to a particular subspecies like the British honeybee. But actually, it's usually better not to identify the honeybee to subspecies, because even within a single hive, you can get considerable color variation. So rather than overinterpret this color, it's best to just, at least in Oregon, to assume that they're all European honeybees. And European honeybees tend to be a hybrid of different so-called subspecies or different populations from various parts of Europe, including but not limited to Italy and the Balkans and so forth. And so these honeybees, which are essentially hybrids, are roaming around. And in the New World, I often prefer just to call them the Western honeybee, which is an umbrella inclusive name for the entire species.
Oh, I see. So I guess if you were in Europe and there still were some pure populations, that would be a value. But here in North America, everything is so mixed up. There's been so many introductions of so much interbreeding that it really just kind of calling it Western honeybee is really the accurate way to call them.
Well, it's even more complicated than that. So what people need to realize is that there are subspecies, which, as you said, are best identified in their native range. So if you're on the Iberian Peninsula and you see a very dark honeybee, you can probably more or less safely call it subspecies iberiensis. But if you're in a place like North America where you are dealing with introduced populations, then it becomes more dicey and maybe less reliable to try to attribute any subspecies identification. So in other words, the subspecies are better used in their native range with native populations if you can somehow verify that.
Okay, but, so okay, we have a lot of color variation. So how do you tell these guys apart from other bees?
Yeah, so just moving on a little bit more. So European honeybee is a name that's used for a grouping of subspecies and hybrids that are the managed honeybees that have traditionally been used across North America. And then we have the African subspecies, which has also been called the Africanized honeybees, or the killer bee, is invading north from California, or invading north into California, but Oregon is too cold for it. So it's pretty safe to call the Oregon bees European honeybees if you want to as well.
Oh, good tip.
But there's only one species found of honeybees in the entire New World. So I think users need to be aware of that. So if you have any sort of a stinging honeybee, it's going to be this species. So how do you tell it? Some distinctive features are hairy eyes, which other superficially similar species wouldn't have. The corbicula or pollen basket, and that's something that you only would otherwise see in the local bees in bumblebees. And usually people can tell bumblebees by their fur coat and colorful pattern and hairy tail or abdomen. So having the pollen basket in the workers. And then probably the best way to verify your honeybee is going to be the wing veins, which are these lines in the wing. And the honeybee has a very distinctive pattern where the marginal cell, or the enclosed area on the anterior margin of the wing, is really long and narrow, almost like a stretched out hot dog or sausage or something like that. So it has a quite different wing venation from other species. So even if you don't want to learn all the technical aspects and the terminology and the homologies, you can still Google wings of honeybee and learn the pattern, and then using your brain's amazing pattern recognition, you can identify if you have a honeybee or not. So I would say the wing veins is the most reliable thing.
And I suppose with wings, it's hard when they're flying, but your picture is going to freeze it, and and I see a lot of these on iNaturalist. You can see the wing veins really, especially with some of these phones have excellent magnification. If you had the whole bee in the frame, you'll be able to see the wing veins.
So actually, the orange and black striped pattern is fairly distinctive. Also, the heart-shaped face where the eyes are converging below. And so basically the head is rather narrow below. That's something that you find in honeybees. You can also see it in some native bees like Calides. And in fact, these Calides native bees are the ones that are closest in shape in some respects to the honeybee.
Mm-hmm.
So they have sort of a conical shape to the abdomen. So when honeybees are misidentified, often they're confused with the Calides. But the wing veins, the patterns made by the lines in the wings are really quite different between these groups. So I would focus on the hairy eyes, the pollen basket in the workers. In the case of the drones, you would have really giant eyes, so they almost look like flies. So the drones are very distinctive as well. The most important thing is not to overemphasize the color pattern, because as I said, it can vary. And it's also very helpful when you're trying to learn to identify bees to look very closely at the details. So rather than just think of the honeybee as orange and black, look very carefully at how most of the abdominal segments may be orange, but then the rim of them may be black, and how they may have pale white hairs at the base. So in general, it's very useful to note not just what the colors are, but what segments the colors are, and whether they're on the apex or the distal or tip of the segments. And then furthermore, whether you're looking at the bare integument or skeleton of the bee, or whether you're looking at hairs. So if you look at a honeybee very closely, or an image of a honeybee, you can see that the orange is actually the skeleton and the black is the skeleton, but this yellowish whitish stuff that you see towards the tail of the bee is hairs. And when you know that it's hairs, then you can understand when it's been worn. You know what I mean?
Mm.
Hairs will wear off, but the skeleton will not wear off. So the more you get into the details of understanding what parts are making the colors, then you can account for variation and wear, for example, abrasion of the hairs, and you can understand much better what's happening. So I would suggest, not just for the honeybee, but for all the bees, that people look in a little bit more detail. Instead of the pattern as a whole, try to break it down by segment. Look at each segment of the honeybee abdomen and see which parts are which color, and then note which colors are made by hairs and which colors are made by the skeleton. Does that make sense?
It does make sense. And I guess one thing I just wanted to put a finer point on, because I do think the first feature is really indicative of honeybees, and you don't always see it, but it is, because not all bees are collecting pollen, but when the pollen is mounding up on that pollen basket, it is quite distinct. The shape of the pollen, the way it rests in that basket, is only really seen in bumblebees.
And also in bumblebees and honeybees, yes. These are the corbiculate bees that have the basket for carrying their pollen. So just to be clear, for those not so familiar with bees, the majority of bees carry their pollen in a brush of hair. And the brush of hair is often on the hind legs, taking the place of this pollen basket, or in the megachilid bees or leafcutter bees and wool carder bees, it's going to be a brush of hair under the abdomen. So bees have different ways of transporting their pollen, and that can be a very useful identification tool to separate the major groups. Okay,
well, that sort of is a good segue to in the top 10 bees that we've seen in Oregon. So the honeybee, there is 770 observations, but ahead of it is a bumblebee. And in the top 10 bees, in terms of frequency of observation, 5 of them are bumblebees. How on earth do we tell them apart?
Yeah, so the bumblebees are very difficult to tell apart because they tend to converge on sometimes very similar or nearly identical color patterns. So as I was mentioning with the honeybees, it's very important to pay attention to the details of the structure and not just look at the generalities of their part yellow and part black, but to see which individual segment is bearing the color. So is the yellow on the first abdominal segment or the second segment, and things of this nature. So it turns out that the most commonly observed bee in Oregon is actually quite difficult to identify, even from specimens, and that's Bombus bosnicenskii. And this is called the yellow-faced bumblebee, and having the yellow face is very helpful to distinguish it from the 6th most common species, which is the California bumblebee, because the California bumblebee has a black face. The hairs on its head are black, and this gives an appearance of an entirely black face and head, contrasting with the yellow forehead and face of the yellow-faced bumblebee. So right there, we can rule out Bombus californicus, but the problem is there are at least 2 other species with a very, very similar color pattern. And one of them is mostly found in coastal Oregon, which is called Bombus caliginosus. I've called it the fog belt bumblebee, but there are other common names for it. But basically, it's nearly identical in general pattern to the yellow-faced bumblebee, and you have to do a very detailed study under the microscope, or look at some very subtle features like length of the coat, or look at the pattern of yellow in the females under the tail, which can be really hard to photograph in order to reliably separate that. So long story short is that the Bombus vosnesensky, even though it's the most common species, can actually be a very serious identification challenge, even to the most experienced experts. And one thing I want to emphasize is the importance of distribution. So if you're in Eastern Oregon, especially far Eastern Oregon, a bee of this sort is not going to be Bombus caliginosus because that's a coastal species. Instead, it's much more likely to be Bombus vosnesensky. But then you also have to consider a less commonly seen species called Bombus van. In that case, the yellow of the tail is on the 3rd abdominal segment as opposed to the 4th abdominal segment in Bombus vosnesensky. So what this really means is that even though this bee is very, very common, it takes a great attention to detail and some comparative experience, and also a knowledge of distributional patterns and local occurrence in order to reliably identify this bee.
Well, one thing I just want to quickly mention, because I imagine people are trying to follow this in their heads, but we will have the link to this list in the show notes. So you can, what I would advise is, as you're listening to the episode, just bring this list up on your tablet and you can follow along. The nice thing is if you click one of those bees, it'll show you multiple images, range maps, and also phenology of when the bees have been found in the area. So it's a very, I would really recommend at this point, if you haven't done this already, open up that link and follow along as we're talking.
Okay, just to clarify how you open the link, you go to the World Wide Web, iNaturalist.org, and then hit the Explore tab, and then fill in bees for the taxon, and then the location would be Oregon, and that's how you make this link. And then if you click on the 116 species, if you click on the tab that mentions species, then it will pull up the species in order of most numerous to least numerous in terms of observations.
Fantastic.
So you can also click on the observations tab and get a list of observations. There's other ways that you can view it, but the species tab is very useful.
Okay, great. We will have it there just in case but I think you can create these lists anywhere you are. If we have listeners in other states, they can use that same workflow to generate a same list for their state. This is fantastic.
Yeah. Mm-hmm. So it creates a sort of de facto state list of not all of the species, because there are hundreds of other species in Oregon, but it gives you a list of those species that are commonly observed and identified.
Okay, cool. All right, so we've talked about so given that the yellow-faced bumblebee can be confused with the fog-belted bumblebee, and I know Bombus caliginosus will find into the Willamette Valley, not just on the coast, although but not in Eastern Oregon. How should we, when we fill in like what bumblebee we think this is, should we just leave it at Bombus, or how would you prefer to see this material classified when it's put into iNaturalist?
Yeah, that's a really tricky question. In general, for me, if you're not sure how to separate it from Bombus californicus, I would simply call it Bombus. But if you're sure that it has a yellow face and is not Bombus californicus or one of these other more distantly related species, then you can call it a Pyrobombus, Pyro Bombus, which is a subgenus that encompasses all of the lookalike species.
So it's a more general category that gets everything. Okay, great. That's excellent.
So this was actually a lengthy debate and discussion on the biodiversity portal Bug Guide, and as a group we collectively decided to have a category of Bombus vosnesensky or caliginosus. I think it's actually presented the other way around, but anyway, it's an either or category, and we categorize those species. So we get into a bit of a different site culture, where at Bug Guide the culture is, in these doubtful cases, to create what we call a no taxon, or something that's not a formal taxon, but is a either or category. And then you can identify them to that level and accumulate images there. And whereas in iNaturalist, they tend to prefer to stick to the formal taxonomy. And so if you're relying only on the official formal taxonomy, then the most precise you can get to is the subgeneric level, Pyrobombus or Pyrobombus if you prefer.
I suppose it's not the end of the world if somebody, I know it's not Californicus because I see it's got yellow hair on its face, and you put it as Vosdesensky. A good taxonomist is not going to trust that probably. You're going to I want to check this a little more carefully.
Well, that brings up a whole nother issue, because these portals are accumulating a lot of data, but before they can be used for science, somebody has to adjudicate that they're fit for use. And I may be biased, but in my opinion, it's the people who have engaged the most with making the observations, making the identifications, curating these sites. They actually understand how these records were assembled and can probably be in a good position to know where the problems are going to lie, what the fixes are, which identifications can be considered validated, and which ones are just a first pass or an approximation. So I think it's very important when these data are used that people not just mine the site, but they need to know not just the data, but the metadata, how these data were assembled, what makes them fit for use or not. And so if people just download the data and mine them and run a bunch of stats, they can come to very much the wrong conclusion. But if you have somebody who deeply understands the data, they can do things like put geographic filters on the records. They can look closely at various criteria of data quality and decide which ones can be used with no problems, which one can be used with caution, and which ones have to be quarantined or redacted to a lower level. So
okay, that's good.
It's actually very important.
Okay, so we've got a yellow-faced bumblebee. We gave it a good thorough treatment, and you were talking about we have another bee that has similar banding patterns, but it has black hairs on its face. Tell us about some of these other bumblebees and how we could tell them apart.
Okay, so there's a bumblebee that's very, very commonly seen in Oregon called Bombus melanopygus. And Bombus melanopygus, the specific epithet means black tail. And so the common name is, remarkably enough, black-tailed bumblebee. And what's very, very interesting is that there are multiple color forms of this within Oregon. So there's a form that's very common in California that has a mostly yellow thorax and then mostly black abdomen with yellow at the base and at the tip. And this is very common in the sort of drier, more southern parts of Oregon, extending into California. But then in the northwestern part, and the more northern and western parts of Oregon, then you see a very different color form that has a grizzled black and gray, or black and pale yellow thorax for the most part, with yellow at the end, and then much of the tail is red. So basically you have a red form and a black form of several bumblebee species occurring in Oregon. And so you need to learn both forms and consider both possibilities to identify this bee. So even though there's only one species, Bombus melanopygus, you have to, when you're doing visual identifications, you actually have to learn two very different color forms.
I suppose for people up in the north of the state, like Portland, they're only going to, for me, it's the most common bumblebee in a birdhouse. I get called every year early in the spring. Bumblebees have moved into my birdhouse. It's always melanopygus. But as I've moved into, like, if you go to Medford or Ashland, you start to see both. Like, I think as you head towards the south of the state, you really are in a bit of a pickle.
Yeah, so I think that this highlights the fact that the knowledge of the local fauna and the distribution of these bees is very helpful. So what I would suggest people do if they're new is maybe to filter the species list not to Oregon but to their particular county, for example.
I'll be darned.
And then if you filter to the county, then you can see what color forms you're likely to see in your area.
That's a great tip. That is awesome. Okay, cool. Okay. So what else do we have for bumblebees?
There's a species called Bombus mixtus, which I'm going to talk about that a little bit. In this species, the tip of the tail, there's a few segments at the tip of the tail that tend to be orange or red, or some mix between red and orange. And the thorax is a mix of black and yellow, and then sort of a grizzled mix of black and yellow hairs. And so that's the name mixtus, refers to the mixed hair color.
Okay.
But the common name of fuzzy horned bumblebees refers to a character of the male, where at the base of the antenna or the flagellum, it's going to have dense patches of hair, which can be hard to see, especially in images, but it's a distinctive feature. So this brings up the point that when you're identifying these bumblebees, and also bees in general, you have to account for pretty significant sexual dimorphism. So the males have their own characters, and they often look considerably different from the females. So you can't just learn the species as a whole. You have to learn to identify the males from the females separately.
Okay. The next bumblebee that we have is, a bee that's pretty distinctive. Tell us about the brown-belted bumblebee, but also, I guess we have a look-alike that's rampant in the north part of Washington now. Tell us a little bit about this bumblebee.
Yeah, so in the eastern North America, so eastern United States and Canada, you are going to see a lot of bumblebees that have a more or less all yellow thorax, and then the first one or two segments of the abdomen are going to be yellow. And this is a Mullerian mimicry pattern that we typically see in these eastern bumblebees.
Oh.
But there's one species called Bombus griseocolus, which is very common across the western North America as well, especially to the north and in the mountains. And some distinctive features of this is that the female, the workers and the queens can have, especially the workers, can have a brown belt. So at the base of their second abdominal segment, rather than having yellow or black as in the other bumblebees, they have a distinctive brownish tinge to the hair patch. But in Oregon, until recently, it was relatively easy to identify this bee because it was pretty much the only species that had this eastern sort of pattern. But what's happening now is that the greenhouse industry has brought the eastern bumblebee, Bombus impatiens, also called the common eastern bumblebee, have brought that in outside of its native range. And it's been accidentally, or not accidentally, but it's been unfortunately introduced into British Columbia, and likely from there has spread down into Washington and maybe making its way still further south. And so we've sort of created an identification problem in the Pacific Northwest by I would say irresponsibly introducing the most common model species for this mimicry complex into an area where it's not supposed to occur.
Interesting.
So the way we can tell Bombus griseocolus is we have to look in detail at various things like the hair coat, how long are the hairs, how shaggy are the hairs, are the black hairs found on the top of the head or pale hairs, and we can also look at whether the pale hairs extend to the second abdominal segment or not, and we can also look at wing color. So long story short, we need to look into details of the pattern. We don't want to look at the generalities of the pattern. And then finally in the males, these are noteworthy because the males have large eyes, really big eyes. And so the large-eyed males can be confused with some other species in Oregon, such as Bombus rufocinctus, which also has the large eyes. But that is going to have more extensive pale hairs on the second abdominal segment.
Well, that's good to notice. Good to note, and also I think we do have listeners in Washington, and there is a good, I think, citizen science project. Chris Looney with Washington State Department of Agriculture is helping track these bees. So keep your eye out. I think this is another great thing with iNaturalist. It will allow us over time to track some of these newly introduced bees as they move. Okay, so let's take a quick break. And then we'll deal with all the non-bumblebees and non-honeybees. Okay, we're back. So we've got well, let's see, 1, 2, 3 bees left, and they all look a little different from one another. Oh no, 4 bees. We got 4 bees left. How do we? I guess I don't know how you want to deal with this. The next one in line, or
Yeah, so why don't we say that there are some cases where it's very difficult to identify bees to species. And one of these is a group called the metallic sweat bees in subgenus Dialictus. So these are the tiny sweat bees that are found in very large numbers across most of the world. And here you often have a dull metallic green head and thorax and a more or less blackish, but sometimes greenish abdomen that's more or less covered in hairs, especially towards the tip. And these can be very difficult to identify, both because there are a lot of lookalike species, but also because they're really small in size. So a lot of the photos just don't have the sufficient resolution of detail to identify them. And then furthermore, it's only been very recently, actually in 2013, that there's been a taxonomic revision of these species that sorts them out technically for scientists. So even in terms of identifying them from specimens, we're still at early stages. We can use the revision of the Canadian species to identify most of the Pacific Northwest bees in this group, but it's a big challenge even for the most experienced taxonomists. So in this case, we're identifying them to a form or a complex of many species, and maybe in the future, when people take higher resolution photos and when people become more familiar with the species found in this region, we can sort them out more finely.
So okay, and this complex, this whole, I guess the complex is a subgenus, so it's not the genus level. There's a multiple species in there, but it's the 5th most common on our list. So there was 131 observations. So this is not a obscure bee. This is, even though it's common.
Yeah. I would say almost anywhere on planet Earth where you have flowers, except for maybe the most extreme Arctic environments, pretty much everywhere in the world, including where I am now in Asia, this genus is very, very abundant.
Really? Okay.
I would say every single one of your listeners should have it in their garden, 100%.
Oh, tell us a little. So it's a small. Are they always small, like a kind of quarter size of a honeybee? Okay.
Yes, they're always much smaller than a honeybee, maybe about a quarter of the size, as you said. And the technical character that you can note is that their wing veins are often weakened, but this can be very hard to see from images. In terms of images, what you look for is the metallic head and thorax, and then in the abdomen, they often have sort of diffuse hairs, often at the base of the segments and also towards the tip of the tail, covering much of the abdomen. But what you don't see is very well organized hairs at the apex. And that, by contrast, that's something you see in another bee on the list called Halictus ligatus, the ligated furrow bee, which is another very common species in this area. And you identify this bee by the pale hairs at the apex of the abdominal segments.
Oh, I see. It's the hairs there are very distinct. When I look at the Dialictus, there's just some faint hairs on the end, and here you've got like literally stripes of hair.
Yeah.
Okay, fantastic. I guess there, in at number 7, we have another smallish metallic bee. How do you tell that apart from these metallic sweat bees?
Yeah, so these bees we can call the small carpenter bees, and the genus is Ceratina, and the subgenus is Zadontomyrus, which is a bit of a mouthful, but in the Pacific Northwest, these small carpenter bees are more or less all going to belong to this subgenus.
Okay.
And so we can tell them because they're elongate and cylindrical. They have a different body form, and that reflects their different method of nesting. So they're actually nesting in pithy twigs or stems, and so they need to have this long cylindrical body form to fit within those constrained nests, as opposed to the ground nesting Dialictus. So the body form is a tip-off. The wing veins are different, although that can be hard to see. And the shape of the head is different in the sweat bees. Often the head is more narrowed below, and it's a more quadrate or bulkier head in the Zodontomeris, in the Ceratina small carpenter bees.
Okay, great. That's great. Yeah, I can see it. I'm looking at the image right now. It looks like it could fit in a twig. It's like made to go in a twig. It looks like
it's a bit hard to describe, but the body form in general can be helpful. Also, the Ceratina bees will have usually some yellow markings. If you look at the foreleg in particular, right at the base of the tibia, so if you look at the front leg at the very top, there's a pale spot. And in the males and in some of the females, you'll also see a pale spot on the face of different sizes and shapes. So whereas in the Dialictus bees, you will not see these same type of pale markings on the legs and or face.
Okay, well that leaves us with two bees. We talked a little bit about Halictus legatus. Did you want to say anything more about it? It's not.
So Halictus legatus can be quite difficult to identify as opposed to Halictus rubicundus, which is another common species in the region. And so again, like all of these bees, you have to look in considerable detail at the things like the head shape. Halictus legatus has a notably large head with broad cheeks that sometimes even form a bit of a tooth at the lower rear margin. So the large head of Halictus legatus is distinctive. When you see them in life, you can tell them because of their smaller size as opposed to some of the other black Halictus species. So moving on to the next one, the European wool carder is a completely different situation because here we have a bee that is native to Europe that has been accidentally introduced to the New World. And then in the 1960s, it showed up in New York. And since then, it's been spread across most of North America, and it's found very abundantly in people's gardens. And the males are notable for being extremely aggressive, and they'll chase off any other visitors other than a female of their species, and they'll even attack and kill honeybees by stabbing them with the spines at the tip of their tail. So the males don't have stings because they're male bees, but they do have spines that they employ as a weapon. So this is a very interesting bee. And it turns out that in Oregon, there are numerous native species of the same genus Anthidium. But unfortunately, as is often the case, the exotic species, which is not supposed to be in the New World at all, is much more numerous than any of the native species, at least in our gardens and in our photographic documentation.
Well, I think those are two ways that I sort of come to think of them is you see the males patrolling, and they're a hefty, large bee, and they're usually around something with some kind of tubular flower, lamb's ear or catnip or something like that. They really don't go. You would never see them on an aster or something. They're really kind of specific to their. I see them on bird's foot trefoil all the time.
So the males are defending different types of flowers. So some of the flowers that the males are defending are flowers that are visited by the female to scrape the trichome or plant hairs that they use for nest construction. So the females get wool by scraping the leaves of these or the plant parts of some of these flowers.
Oh, that's the wool.
That's the wool.
Good.
So the wool is scraped using the mandibles of the female from these different plants. But then the females are also visiting long tubular flowers to get high quality nectar from these nectar sources, but they're visiting still some other plants for pollen. So the females will actually be visiting plants for three different purposes.
Really?
And the males will be guarding all three of those floral resources. So it's a pretty complicated situation. But one thing to note about the flowers is that there are the native species will often be associated with different flowers, like, for example, Phacelia. And meanwhile, there's another exotic species called Anthidium oblongatum, which has a slightly different color pattern. And this one is associated with other plants like sedum and So they do overlap and they do occur on the same plant, but their preferences are not exactly the same. So you can use host plant as a clue to suggest what sort of an Anthidium you may be dealing with. And just going back to how do we recognize an Anthidium, you can look at the very distinctive yellow and black colors, the spines at the tip of the tail in the male, and then the females have large, many-toothed mandibles that they use for scraping the wool from these plants.
And the yellow is really, most people wouldn't associate it with the bee. It looks like yellow jacket yellow. It's a very bright yellow.
Yeah, so I think it's fair to say that these Anthidium are mimicking some yellow jackets that live in their native Europe.
Uh-huh.
So it's an interesting case where sometimes these exotic bees will look exotic and look strange because they're actually mimics of species that don't occur in our area. Although we do have yellow jackets, but I haven't gone into this in detail, but most likely they're a very close match to a European species of yellow jacket as opposed to our North American ones because they evolved this color in Europe, not here.
Well, fantastic. Thanks for taking us through the first 10. We'll have to have you back on again just to take us through the next 10. But this is really good. I think these are some of the most common bees. And the tips that you provided on how to use iNaturalist so we can generate some more records for the state, I think those are – I'm going to go back and listen to those. That was really great.
Okay, well, thanks for giving me the opportunity to discuss these things. I'm really excited about seeing the quality of the images, improving our online field guides and reference material. But in the case of Oregon, I'm extremely excited about having coverage for parts of the state, like remote areas of eastern Oregon, where historically there's been very little collecting, or where the collecting was done long ago, and now we can bring in new observations. And I want to bring up a couple of points that is important in Oregon and other places, is that there are many places like, say, Crater Lake National Park or any national parks where in the Pacific Northwest where it's difficult or impossible for the general public to collect for obvious reasons. But by doing these, by using images, people can, in the course, whether they're out birding or doing nature photography or looking at wildflowers, they can actually go to these protected areas, wildlife refuges, and they can document bumblebees and all of these other bees in a non-destructive way. And I think it's a solution to what otherwise would be a very intractable problem, which is the areas where we most want to get an inventory of bees and to monitor their changes are some of these protected areas, but those are the ones that are off limits to so-called casual or opportunistic collecting. And so this is one of the main reasons why I devote so much time to this, is because I think it's crucial for bee conservation to get inventories in places like Eastern Oregon and to continue to monitor areas that are not accessible to your average collector. I hope that's clear.
Well, on behalf of many bee iNaturalist users, thank you so much for all your efforts and getting and all the other platforms as well. This is really, I think, expanding the scope of what regular people can contribute to science. Thank you so much.
Okay, thank you for the opportunity to discuss these things.
Thank you so much for listening. The show is produced by Quinn, Sin, and Neil, who's a student here at OSU in the New Media Communications program, and the show wouldn't even be possible without the support of the Oregon Legislature, the Foundation for Food and Agricultural Research, and Western SARE. Show notes with links mentioned on each episode are available on the website, which is at pollination.podcast.oregonstate.edu. I also love hearing from you, and there's several ways to connect with me. The first one is you can visit the website and leave an episode-specific comment. You can suggest a future guest or topic, or ask a question that could be featured in a future episode. But you can do the same things on Twitter, Instagram, or Facebook by visiting the Oregon Bee Project. Thanks so much for listening, and see you next week.